Hello, my name is Tilo Kunath, and I am a professor at the University of Edinburgh. And it's a pleasure to give you this talk today. And thank you to LabRoots for inviting me. So my background is developmental biology and stem cell biology. I did my postdoc in Edinburgh, at the University of Edinburgh, and now I'm a group leader at the Institute for Regeneration and Repair, as well as the UK Centre for Mammalian Synthetic Biology. And today I'm going to tell you about a toolkit that we've been developing for dopaminergic progenitor cell production from pluripotent stem cells, such as iPS cells and ES cells. And I'm going to tell you about work from students in the lab, so Yishi and Craig that you can see here, also from a postdoc, Nicola Drummond. And my declarations of interest, I'm going to show you some work that we did with a company called UCB, and that co-funded some of the work, and also a company in Cambridge called Asymptote, now known as Cytiva. Okay, so let's get started. So I got into this area about 10 years ago with a really fruitful collaboration with Professor John Hardy at UCL. So he had access to fibroblasts from this quite famous Parkinson's kindred in Iowa. So Henry, Patrick, and John provided 2 skin fibroblast samples from this mother-daughter pair. So Mike Devine and I used the fairly new Yamanaka method. This is over 10 years ago, 2009, 2010, to make iPS cells from these 2 individuals. So we made 30 lines from the mother. So the mother harbored triplication of the alpha-synuclein gene, and this is what was causing Parkinson's in their family. And we call her and her iPS cells AST, and this is short for alpha-synuclein triplication. And we also made a number of iPS cell lines from her daughter, who did not harbor the triplication mutation. And we call her lines normal alpha-synuclein, or NAS lines. So again, I'm not going to get into this paper. It's published quite a while ago, but I just wanted to highlight one of our major findings back then, because we made so many cell lines from 2 people. We made 40 lines from 2 people. We really appreciated quite a while ago the variability between different iPSC lines, even from the same person. This is a theme that comes up again and again. I think it's much more well appreciated now, but something we need to take into account, and that I'll discuss in my talk. So in addition to iPS cell lines, we work with a lot of embryonic stem cell lines. So the UK has the largest collection of clinical grade ES cell lines in the world. So they are about 38 lines that were derived over the last 15 years. And we published the molecular karyotype of 25 of them a few years ago. So one of these cell lines, RC17, has actually begun its clinical trials in patients. So a patient only a week or so ago in Lund has been transplanted with dopaminergic progenitor cells from the RC17 cell line. I'm going to talk about these dopaminergic progenitor cells in my talk. So we work with a lot of cell lines in my lab, maybe close to up to 100 cell lines we've been working with. We grow them all the same way. iPS and ES cell lines we treat identically in terms of our culture conditions. The only difference between these 2 types of cell lines is where they come from. So the ES cells are all derived from donated blastocysts from IVF clinics, ours from within the UK. And the iPS cell lines, of course, come from consenting adults, normally derived from skin or blood. So we grow them on a recombinant matrix, laminin 521. Our favorite medium is iPS brew from Miltenyi. And they all have their idiosyncrasies. They all have these slight differences, but they all express pluripotent transcription factors such as NANOG. But what we've been spending vast majority of time is turning these stem cells, iPS cells and ES cells, into useful cell types for either transplantation or for disease modeling. And we also do CRISPR engineering of the iPS and ES cell lines, so I'll show you some examples of that as we go on. So I'm going to go through the dopaminergic differentiation in great detail. I think it's interesting and important, and it pulls on some of my developmental biology background. And the main point of this slide is that to show you that it takes time. So getting neurons from iPS cells takes months. To get them really functional takes a lot of time. But transplantation-ready progenitors doesn't take that long. So transplantable cells can be made from iPS and ES cells in just a little over 2 weeks. Okay, so how do we turn iPS and ES cells into transplantable cells or into neurons? So we have to take sort of cues from the embryo, and it's been published now by several groups that the progenitors to substantia nigra neurons or dopaminergic neurons is an embryological structure called the floor plate. And the midbrain floor plate here circled in orange is in the mouse expresses certain transcription factors such as LMX1A, FOXA2, and it's in the ventral part of the neural tube, so the bottom part of the neural tube. And and as these progenitors mature and exit the neural tube, they form these nascent dopaminergic neurons. On the paper on the left, these are expressing the enzyme that's needed to make dopamine, tyrosine hydroxylase, shown in green. So if we can make midbrain floor plate in a dish, then your chances of making the dopaminergic neurons is very high. So how do we do that? So again, we take advantage of the known signaling cues that the embryo, the vertebrate embryo, uses to make this tissue in vivo, and we apply that to our in vitro settings. So the first sort of manipulation we need to do to the ES or iPS cells is to inhibit all SMAD signaling. This is called dual SMAD inhibition. We use 2 small molecules to accomplish this, and this turns the ES cells and iPS cells into anterior dorsal neural tissue. So where the bracket is showing here, the telencephalon above. So layering on top of the dual SMAD inhibition, we add a very high level of sonic hedgehog activation. And this dorsalizes the tissue, this ventralizes the tissue. And as shown by the green arrow there, it's now a ventral part of the neural tube, but it's still very anterior. So it is still forebrain ventral tissue. So to posteriorize this tissue, we need to add a certain level of Wnt, a precise level of Wnt, or a tuned level of Wnt, I should say. And this posтерiorizes the tissue from a forebrain identity to a midbrain identity. So we cannot add too much Wnt because then it'll turn this floor plate tissue into hindbrain. So this is the hardest part of the protocol to get right, as I'll demonstrate in some of the data I show you. But this tuned level of Wnt is critical to get midbrain floor plate. Hindbrain floor plate or forebrain floor plate will not give you dopaminergic neurons. And the last level of signaling that we add is FGF8. This is to mimic this organizer tissue shown by this red stripe. The isthmic organizer, which secretes FGF8, it also helps pattern the midbrain floor plate tissue to get the dopaminergic neurons. Okay, so how this looks like in a sort of linear time fashion is shown here. So the iPS cells and ES cells are given dual SMAD inhibition, a high level of sonic hedgehog, and a tuned amount of Wnt activation from the beginning. So we activate Wnt pathway with its GSK3 beta inhibitor, CHR99021. And that's present for first week and 2 days, and this is all removed, and then FGF8 is added for the next 7 days. The rest of the factors are just important for neuronal health and neurotrophic, so those are not important for getting the midbrain floor plate. So as you can see, again, it takes time. There is 2 lifting and replating steps, and you can freeze them at these days, day 11 and day 16. And as I showed at the beginning, they're transplantable by day 16. So as we're going through this protocol and turning these stem cells into floor plate tissue, we can check by markers, are they really making floor plate tissue, midbrain floor plate tissue? And the transcription factors I showed you earlier, LMX1A, FOXA2, you can do immunostaining for, and if the protocol is going well, you should see nice double positive LMX1A, FOXA2 cells by day 11. By day 16, you should see a third transcription factor coming on that's very indicative of midbrain floor plate called engrailed1. And again, when the protocol is going well, you'll have really nice engrailed1 expression by day 16, co-expressed with FOXA2. A fourth marker is a protein called corin. And this is a useful and interesting protein because it's a cell surface marker. So and it's also expressed exclusively in the floor plate. So you can see in this image from Ono et al., this is staining for corin just in the floor plate. But to the left and right, where the dopaminergic neurons are emerging, corin is downregulated. So you can use this marker for FACS. You can do flow cytometry at day 0, meaning before you start the differentiation, where very few cells are corin positive. At day 16, at the height of making midbrain floor plate tissue, the vast majority of them in this experiment are positive, where we can get 99% of the cells expressing corin on the cell surface. And as you continue the differentiation to day 25, you can see that the cells are becoming less positive for corin, and they're shifting to the left. And this is because they're turning into nascent dopaminergic neurons and downregulating corin at this point. So if you continue the differentiation, and here's an image from day 45, it's a whole well image. So if the floor plate tissue was really pure at day 16, and you continue the differentiation, you'll get something like this, which is essentially a neural network of dopaminergic neurons expressing the enzyme that's needed to make dopamine tyrosine hydroxylase. These neurons are morphologically like neurons. They have not formed synapses by this point, and they're not electrically active at this point. However, if we keep going, so now we're going over 100 days of differentiation, they do have some beautiful spontaneous activity that we published a few years ago with Sergei Stanislav. Also in this paper, we show that these neurons are making a vast amount of dopamine. So there's a lot of dopamine being released in the medium just spontaneously. If you differentiate them beyond day 80, I'd say beyond day 75 to 80, you can start getting this nice electrical activity and dopamine production. Okay, so showing you all the rosy stuff, all the good stuff. So as I mentioned at the very beginning, we've worked with a lot of cell lines in the lab. And one of the themes that comes up again and again is that different cell lines have different propensities for differentiation into all lineages, but in particular into dopaminergic differentiation. So here's an experiment from Nicola Drummond in the lab, taking 2 different cell lines, MAN12 from Manchester and MasterChef7 from Sheffield, differentiating them in our standard protocol, and one cell line is reacting very well, lots of good differentiation. Another cell line, the differentiation is not as good. There's nothing wrong with MAN12. MAN12 is a very good cell line, karyotypically normal, and has all the pluripotent expression markers. So it's just genetic. They are genetically different than MasterChef7, and they just react differently to this particular protocol. So either we need to change the protocol for MAN12, or maybe it has a better propensity for making endoderm or mesodermal tissues, and maybe dopaminergic is just naturally not good at. So we don't know the reasons, but this is something that we see again and again. But more problematic is when we have this one cell line reacting differently to differentiation on different weeks or different months. So this is also common, where you can try differentiating one of our favorite cell lines, RC17, and it doesn't always go to plan, where you have the staining for LMX1A FOXA2 is not as beautiful or as homogeneous as you would like. And the corin FACS, which I showed you earlier, I showed you a 99% corin FACS, can in some cases give you a double peak, where some of the cells are the desired midbrain floor plate, but sometimes and some of the cells are not. And this is just an example of 2 CRISPR clones that we had made from RC17, but even the parental cell line wasn't behaving well in that month. But what was really surprising and sort of difficult to comprehend at the time is that very, very small changes in this Cure molecule, this Wnt activator, could fix this variable or heterogeneous differentiation. So this is the compound that we add from the beginning. It activates the Wnt pathway, but changing the concentration by a very small amount, less than 10%. So 1.1 micromolar versus 1.2 micromolar can give you a very different FACS profile at day 16. So you can see a very more or less homogeneous peak at 1.2 micromolar and a more broad heterogeneous peak at 1.1 micromolar. So the sensitivity of this protocol to Cure 99021 is very high. So if we take these 2 cultures at 1.1 and 1.2 and try to make neurons from them, the 1.2 micromolar Cure floor plate cells gave us very homogeneous neuronal population, while the 1.1 that had the broad core and peak gave us a mixture of neurons and non-neuronal cells. There are neurons in there, but they are mixed in with some non-neuronal cells. So we tried to investigate a little bit of why this protocol might be so sensitive, the activation of Wnt. And one of the reasons is that these cells themselves make their own Wnt molecules as they're differentiating. So here are two Wnt that are known to be expressed in the midbrain, Wnt5A and Wnt5B, qPCR from ESLs all the way up to day 11 progenitors. So this is the endogenous Wnt we're looking at. You can see that these are going up in a time-dependent manner. So we asked, are these Wnt signaling onto themselves, so autocrine or paracrine signaling? And we can ask this question by adding in an inhibitor of Wnt secretion. So this molecule, IWP2, is a porcupine inhibitor, and it prevents the release of Wnt from cells. So it doesn't affect the Cure activation. So Cure inhibits GSK3 beta, so it activates the Wnt pathway downstream of the receptor. So when we of course normally never add this porcupine inhibitor. So in the absence of this porcupine inhibitor, with all of the normal goodies in the protocol, in this particular experiment, we got 90% corin positive midbrain floor plate, but 250 nanomolar, or as low as 50 nanomolar, can destroy this homogeneous corin peak. So very small amounts of this porcupine inhibitor, meaning inhibition of endogenous Wnt, can really alter the protocol. So this means that the total Wnt signal that cells are seeing during the differentiation is a summation of the Wnt that we activate artificially with the Cure 99021, plus the endogenous Wnt, probably from Wnt5A, Wnt5B, that I showed on the last slide, combining to give us that Goldilocks level of Wnt activation. And the Wnt5A, Wnt5B, we have no control over of how much is being made and how much is being secreted. So getting that fine-tuned balance of Wnt activation is quite tricky, especially for this protocol, because of these endogenous Wnt that we cannot control. So we have a couple strategies to make our lives easier. One of them is a fairly big and long project that we've been working on for a number of years that I'll just give you a bit of a taster of. But we are monitoring our differentiation in real time from the earliest stages using the conditioned medium. So I showed you transcription factors that change over time. Cell surface proteins, of course, will change. The transcriptome will change over time. But also the molecules that the cells secrete into the medium will also change during the differentiation. So we've been using this secretome information to non-invasively monitor the differentiation. I'll just show a couple of examples of that. Well, I'll show you this one particular example, which has been really, really fruitful for us. So this is a microarray expression profile of differentiating stem cells into dopaminergic neurons from Lorenz Studer, published over 10 years ago. And there was this unusual molecule that appeared on the list that I didn't recognize. So trefoil factor 3. So this protein, which was sort of being upregulated, similar to LMX1A-FOXC2, was not a transcription factor, but a secreted protein. So to make a long story short, we found that this protein is released into the medium to very high levels during the differentiation. So this is data from ELISA. This is a TFF3 ELISA looking at conditioned medium from stem cells. So day 0 would be undifferentiated cells, up to day 25 dopaminergic neurons. You can see there's a time-dependent increase of this protein in the medium. But what was really helpful and interesting with that is that the secretion of trefoil factor 3, or TFF3, we could detect as early as day 4. And this detection at day 4 was predictive of a good differentiation or a bad differentiation. So the top are three conditions with sonic hedgehog. The high level of sonic hedgehog is our normal condition, and it's a day 11 immunostain. But condition medium from day 4 on the left and day 7 on the right, we can see differences on TFF3 already, predictive of how the floor plate will form at day 11. So this is really early information to know, are we on a right trajectory? Is this going to be a good differentiation or not? So, and this data is looking at the level of Cure. So this is the molecule that needs to be not too high or not too low. And again, as early as day 4 and day 7, we can see that the peak of TFF3 expression is predictive of how nice or how homogeneous the cells will be in the future at day 11. So this has led to quite a large project with Asko Morizane, funded by the MRC in the UK and the AMED in Japan. And we've discovered a number of other novel secreted markers using mass spectrometry and other techniques that we've been combining into multiplex systems to generate essentially a tool to tell us are cells on a good differentiation path or not. And we've multiplexed them using this technology, Luminex. And I don't have any data to show you now, other than to say that we've made 2 versions of this Luminex platform, combining both positive markers that include TFF3 and some negative markers of cells that are undesirable, that we don't want to see coming up. So these different platforms of Luminex have been very helpful for us to sort of monitor the differentiation and know if we're on the right trajectory or not. So the second thing that we've been doing sort of just naturally over the years is that we've been freezing the cells at the progenitor stages and doing a lot of quality control of these progenitors when they're frozen. And the useful thing about freezing at day 11 or day 16 is that this is beyond the time when we would add this problematic or finicky molecule, Cure 99021. We spent actually quite a bit of time perfecting the freezing of this cells after what we call the difficult stage of the protocol. I think, yeah, it's just we're very now familiar that if we can get them to a good pure floor plate stage at day 16, the rest of the protocol goes well, and the expertise needed is actually very, very little to do this if you get your hands on some good floor plate cells, some good frozen floor plate cells. So we worked with John Morris and Peter Kilbride in Cambridge to really perfect the maximum recovery of frozen cells at day 16. There's a lot of freezing data in this paper, but the main message is that we can now we are converting our iPSC banks into midbrain dopaminergic progenitor banks. And these banks we're using internally, but I'm going to also show you 2 examples of collaborations that have allowed us to do some really exciting work, because we can send around these frozen progenitors to labs that essentially don't have the expertise to do the differentiation. And yeah, and the main message is that freezing or not freezing does not alter the differentiation path of these cells. So at the top, there are some non-frozen cells that made beautiful dopaminergic neurons, but if they're frozen, it doesn't change the trajectory of the differentiation. Okay, so in the last 5 minutes, I'm going to show you 2 examples of how this has opened up collaborations. So the first one is with the collaboration with Mariah Lelos and Emma Lane in Cardiff. So their expertise is in animal models, transplantation models of Parkinson's using rat models. So we sent them day 11 cryopreserved midbrain dopaminergic progenitors, and they grew them for 5 days in Cardiff and transplanted into this well-recognized Parkinson's model, the 6-hydroxy unilateral lesion model, where the dopaminergic neurons are lesioned on one side of the brain, and the same side of the brain is transplanted with progenitors a few weeks later. So this is some staining of cells that were grown in Cardiff. This is now Corin immunostaining, but you can see it looks fairly homogeneous. And here's an example of one of the rats that was transplanted with these cells 10 weeks after transplantation, stained for tyrosine hydroxylase. The left side is the unlesioned side, so you can see the fully intact striatum. On the right side, you can see the 2 deposits of human dopaminergic neurons. So 10 weeks is fairly early into the experiment, but this is a different rat at 20 weeks post-transplantation. You can see the staining here is for STEM121, a marker of human cytoplasmic. It's an antibody that recognizes human cytoplasmic antigens, and you can see there's a lot of human neurons fully innervating the rat striatum. And if an adjacent section to that shows that most of these human neurons are dopaminergic, meaning they express this tyrosine hydroxylase enzyme. And there's a phenotypic assay that can be done on these rats. It's called the amphetamine-induced rotation assay, where the animals are, when injected with amphetamine, predictably turn in an ipsilateral direction to their lesion, and this phenotype is stable for the lifetime of the rat, unless the lesion is fixed. And you can see the rats that received this midbrain dopaminergic progenitor graft, at 10 weeks were still rotating when the grafts were small, but at 20 weeks, this rotation phenotype is almost completely dissipated or disappeared. And you can see over 11 rats, it was very, very consistent rescue of the phenotype. So this ability to transfer progenitors to Cardiff for this transplantation, and also Lund, the group in Lund, Malin Palmer and Agneta, also were transporting progenitors to Cardiff, and they could compare these progenitors in their rat models, led to a very interesting and quick publication. So last example I'm going to show you is a fun and interesting project on disease modeling. This is related, I think it is related to Parkinson's, but it may not appear so at the beginning. So we got really interested in this paper published by David Beckham in Denver several years ago. We were trying to determine what the endogenous function of alpha-synuclein is, or the physiological function. And this really struck us that he was suggesting that alpha-synuclein was a neuronal innate immune molecule, and that was important for protecting against virus infection. And this figure panel from his paper showing that alpha-synuclein null mice were highly susceptible to West Nile virus infection was striking. So this is a whole animal null infected with West Nile virus versus wild type, and you can see that the null animals succumb to the virus very dramatically. So we had been deleting alpha-synuclein from our iPS and ES cells using this CRISPR strategy. So we could get homozygous deletions in ES cells using this double nick strategy. And we could also get deleting the alleles from the triplication iPS cell lines that I mentioned at the very beginning. So we had a lot of null cell lines. One of the take-home messages is that deletion of alpha-synuclein does not affect the differentiation. That the differentiation to midbrain floor plate was very efficient in the presence or absence of synuclein, and we could make dopaminergic neurons just fine without alpha-synuclein. So this gene wasn't important for differentiation. So we contacted David Beckham and Yishi, who had done all those knockouts, prepared null and wild type day 16 dopaminergic progenitors, sent them to Denver, and they recovered them and grew them into more or less immature neurons, and then infected with the RNA viruses that they have there. So these are now pure neuronal populations. They made beautiful neurons, again, with no expertise. They don't have expertise in iPSC differentiation, but they were able to take our progenitors and turn them into neurons very easily. And the striking result here is shown on the right, where the null and the wild type neurons are infected with this Venezuelan equine encephalitis virus, which is an RNA virus that is neuroinvasive. You can see the null neurons are highly susceptible to these viruses, and there's a 1,000-fold higher viral titer shortly after infection. So part of the mechanism behind that we published last year. So we showed that the interferon response, an innate immune response, is blunted or muted in the absence of alpha-synuclein. So the interferon inducible gene IFIT1 is induced on virus infection or by treating the cells with this viral mimic, but nowhere near as efficiently as wild type cells. So we think this is part of the answer of why these neurons without alpha-synuclein and the mice without alpha-synuclein are susceptible to virus infection. So how this connects with Parkinson's, I think, is a very interesting question and something that we're working on now. Okay, so I showed you that the tuning of the Wnt activation pathway is incredibly important from the very beginning of the differentiation. We are now have tools to monitor the differentiation in real time using secreted biomarkers, and this is really helping us to fine-tune the protocol. I showed you that cryopreservation is possible, but it's also been extremely handy for us to do a lot of the collaborations I mentioned. And this also for clinical trials, this allows you lots of time to do quality assurance and safety testing. Okay, so this is more or less what I said just now. So tuning the Wnt activation is incredibly challenging and important for the differentiation. So non-invasive real-time markers can improve the consistency of our cell-based products. But again, thinking about clinical translation, it could reduce production costs, because you could stop production if a differentiation. You could stop production quite early if the differentiation is not going well. And the cryopreservation of these poised cells that are essentially ready to go has really helped us internally for reproducible differentiation. But it's also allowed for these fun collaborations that I showed you with labs that don't have the expertise in pluripotent stem cells, or don't wish to set it up. It did take us 2 years to set up the differentiation and to get it going at the levels I showed you. And also for the translation, for transplantation, it will allow time to conduct safety and efficacy assays if you can freeze the cells. Okay, the last slide is just to thank a lot of the people that did the work, and a lot of the funders that have funded along the way. So I mentioned a lot of the names already, but I want to do a special mention to Cure Parkinson's Trust and Parkinson's UK that got me off to start for my research career in Parkinson's. And thank you very much.